Hey guys, welcome to AP Biology or College Biology. We're going to be focusing in on section 5.2, which is all about meiosis and genetic diversity. Please note this is on Unit 5 Heredity. If you haven't done so, make sure to check out our earlier videos related to Unit 5 because that will give you more details as it relates to meiosis, which is a really good foundation if you're planning to take the AP Biology exam. Please note the disclaimer listed below and get started. Okay, so when we're reviewing meiosis, I want you to understand that there is a whole section related to understanding meiosis and meiosis 1 and meiosis 2, and that's section 5.1. If you haven't done so, please review meiosis and mitosis too, which is all in unit 4. The reason why I say this is if you're planning to take the AP biology exam, you should note that oftentimes, just as a little hint, they like to compare mitosis to meiosis, compare and contrast, I should say. So, making sure that you review our entirety playlist. We have an AP Biology Everything You Need to Know playlist, and we also have the previous units from units 1 through 5 up until now, or even later if you're watching this at a later time, and you can review each one of the units and really understand more details as it relates to AP biology for the exam coming out. Okay, now let's get back to understanding meiosis further. So previously, if you remember, we talked about meiosis and meiosis 1 and meiosis 2, which is different than mitosis, which only has one step as it relates and divides. But if you remember, we have meiosis 1 and meiosis 2. And the reason why we have these two steps is because we're trying to produce gametes or sex cells. The purpose of meiosis in general is to create this genetic diversity, to understand that we can carry on genes very differently from offspring to offspring or from parent to child to another child. And that's done through three different ways. The first one is crossing over. Crossing over, we're going to talk about more in detail, but it's this idea that the chromosomes line up and then they switch to eventually get new combinations. Then we have independent assortment, which is the idea that at some point you're going to have random gametes that will be produced with random combination. Sort of like when you play a card game and you're mixing up the cards and you're dividing up the cards amongst your friends, you really have a random combination of cards. And so that's independent assortment. This leads us to random fertilization, which is the act of that these gametes, which include egg and sperm, can be mixed in a variety. You don't have the same egg going to the same sperm. That's where random fertilization comes in. Okay, now let's talk about crossing over. Now, if you remember, in meiosis, we have two steps, meiosis 1 and meiosis 2. In meiosis 1, which is very similar to mitosis, our goal is to take one cell and divide it into two cells. But what happens is they end up having half the chromosomes in each one of these cells from the original cell, and they have different combinations. That's because if you have a pair of chromosomes, remember you get one from one parent and another from another parent for each gene, those are called homologous chromosomes. So, for example, let's say you have a pair of chromosomes. Remember, we have 46 total chromosomes and 23 pairs, and each pair is considered a homologous chromosome, one from dad and one from mom. What ends up happening is during prophase 1, because remember the steps are prophase, metaphase, anaphase, telophase, cytokinesis. This all has to do with the first meiosis. So prophase 1, metaphase 1, anaphase 1, telophase 1. Then you undergo prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. But we're talking about that very first step where, if you remember in prophase, this is when the chromosomes are getting ready by having the nucleus getting ready to disintegrate. What's happening also in this step is you're going to have chromosome combinations that are going to switch up. What ends up happening is the segments of the chromosomes from each parent, so one from mom, one from dad, one from mom, one from dad, they will switch off. So they sort of like line up and not at like in metaphase, but they're getting ready to attach to each other. So one from mom, one from dad. And then what ends up happening is they sort of swap and then they produce a unique variation of new chromosomes that will now line up at the metaphase plate. So this is all occurring at prophase 1 of meiosis 1, and this is when the homologous chromosomes pair to form what are called tetrads. And what they mean by tetrads is 1, 2, 3, 4. Tetrads mean 4. Okay, tetra, 4. What you want to understand is you have four different possibilities essentially, and the genetic material is exchanging between non-sister chromatids of homologous chromosomes. So these are not sister chromatids. They're going to go ahead and swap. And the purpose of this crossing over is to create new combinations of chromosomes so that way you have new alleles and new possibilities that can be inherited in future offspring. And then this leads us to the second way that we can increase genetic variability, and that's independent assortment. Now, remember, we have meiosis 1 where we have the divvying up from 1 to 2, and now you have to think about the second division, that second combination that occurs. So meiosis 2, if you remember, is this idea of taking the chromosomes, lining them at the metaphase plate, and then pulling them apart. Now, in metaphase 2 versus metaphase 1, we essentially have those individual chromosomes that are the new mixtures that happen due to crossing over. And what ends up happening is they'll end up splitting. So you have this split, and then this split, and then this split, and this split. So now you have four different varieties or four unique combinations of chromosomes that weren't exactly the same as the parent. The benefit of this is you have 2 to the 23rd power of distinct chromosome combinations of egg or sperm that one can generate within humans. Remember, we have 23 pairs. So you could have essentially a variety of different combinations. So once again, that also increases our genetic diversity and why parents and offspring don't look exactly the same. Of course, the final way to increase genetic diversity is random fertilization. Remember, we have one sperm, and that is going to compete for thousands of spots for, I shouldn't say thousands of spots. There's thousands of sperms essentially that will try to compete for one egg, one single egg. And so each sperm and each egg carry a unique set of unique combinations. Remember, if you remember in meiosis, they're producing gametes. So this is one combination, one combination, one combination. They're unique. They're different combinations that are competing for this one egg. So the randomness of fertilization increases also genetic diversity. You have two parents that can't produce exactly the same child twice, and you have the variation of egg and sperm. Also, there's something noted as nondisjunction. So, nondisjunction is this idea of genetic mechanism that's related to errors that may exist. So, let's say, for example, in meiosis 1, you don't have proper splitting. You don't have the splitting of the chromosomes in half. Or maybe in the meiosis 2, there could have happened where one egg will have two chromosomes while the other one only has zero chromosomes. That can happen as well. These are all forms of non-disjunction, and that's due to the fact the chromosomes don't divide properly. And this can occur in both anaphase 1 or anaphase 2. If non-disjunction occurs in meiosis 1, all the haploid cells will have an abnormal chromosome count. If it occurs in meiosis 2, only two of the resulting haploid cells will be affected. Non-disjunction can lead to miscarriages or genetic disorders. A common one is trisomy 21, where the 21st chromosome is affected by having an extra chromosome attached to a pair. So essentially, there's three chromosomes that are characterized by the extra 21st chromosome, which is also known as Down syndrome. So that's just an example of how non-disjunction exists. Okay, so let's try to conquer a quiz question. Take a moment, pause the screen here, review the question, and then unpause once completed. Now that you're done reviewing the question, let's review it together. It says, what is random fertilization? Is it A, the process by which one sperm always fertilizes an egg? B, is it a random chance of specific sperms fertilizing a specific egg? Is it C, the fertilization of multiple eggs at the same time? And D, the creation of identical offspring from the same parents? So random fertilization is best chosen by B, which is a random chance of a specific sperm fertilizing a specific egg. Remember, you're not always going to fertilize an egg with one specific sperm. There are a ton of sperms that come from one individual. And then answer choice C is not best because you have multiple eggs, whereas that's not true. It's one egg that's usually competing for multiple sperm. And identical offspring are not random fertilization. Random fertilization is a very specific sperm choosing a very specific egg. So that's the best answer choice. I hope you enjoyed that quick video as it understands and relates to section 5.2 while we hone in on meiosis and the understanding of genetic diversity. If you want to review earlier videos, make sure to check out our earlier playlists as it reviews units 1 through 4. If you're watching this at a later time, make sure to review the videos related to unit 5, unit 6, unit 7, and unit 8 that are planning to come out. Make sure to hit that subscribe button for later videos as well. And as always, make sure to check out these videos for a review for AP Biology or College Biology success. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.